A recent study published in Translational Psychiatry found that exercise can mitigate the negative effects of disrupted gut microbiota on hippocampal neurogenesis and cognitive function in rats. The study aimed to explore whether physical activity could counteract the adverse impacts of gut microbiota disruption on the hippocampus, a brain region essential for memory and spatial navigation. Human brain cells, or neurons, typically stop dividing shortly after birth. However, the hippocampus retains the ability to generate new neurons throughout adulthood, a process known as adult hippocampal neurogenesis. This neurogenesis is crucial for learning, memory, and mood regulation and is influenced by the gut microbiota through the microbiota gut-brain axis, a bidirectional communication pathway. Researchers conducted experiments on 9-week-old Sprague Dawley rats, dividing them into two main groups, sedentary and exercise. Each group was further split into subgroups, with one subgroup receiving antibiotic treatment to disrupt gut microbiota and the other not. The exercise group had access to a running wheel, while the sedentary group did not. 3 weeks into the treatment, the rats underwent behavioral tests to assess cognitive abilities. Afterward, their tissues were analyzed to evaluate hippocampal neurogenesis. The results showed that gut microbiota disruption induced low-grade peripheral inflammation without affecting body weight or running activity. Rats with disrupted microbiota and a sedentary lifestyle exhibited worse cognitive performance and increased anxiety-like behaviors compared to those with intact microbiota. However, exercise mitigated these adverse effects. Disrupted gut microbiota also reduced hippocampal neurogenesis, but this reduction was less pronounced in the exercise group. The researchers identified specific biochemical compounds associated with these changes, highlighting the expression of ethyl 2,4-oxo-4,5-dihydro-1,3-thiazole acetate. The study concluded that exercise partially reverses the behavioral and neurogenic impairments induced by gut microbiota disruption, emphasizing the importance of the gut microbiota in behaviors dependent on adult hippocampal neurogenesis and the beneficial role of lifestyle factors like voluntary exercise. While the findings shed light on the protective effects of exercise on hippocampal neurogenesis, it is important to note that the study was conducted on rats. Although rats and humans share many physiological similarities, the effects on humans might not be identical. Further research is needed to determine if these findings apply to humans.